Hello everyone, my name is Rachel Zadovnik and I'm a first year undergraduate student at Northeastern University, and today I'll be talking to you about mitochondrial diseases and how there are stem cell therapies out there and the research that is being done with them to help treat these incurable diseases. To begin with, the mitochondria is one of the organelles in cells that helps produce adenosine triphosphate, also known as ATP. ATP is this power molecule in the cell that powers all the functions of the cell. So if there's mutation to the mitochondrial DNA, then ATP cannot be produced, and therefore the cell will not have ATP to do its normal functions. Mutations to the mitochondrial DNA can lead to diseases that are heavily focused on muscle weakness and deterioration, as well as vision loss and dementia. So, through the use of induced pluripotent cells, researchers believe that they can find a way to turn this unhealthy mitochondrial cells into healthy mitochondria, with cells with healthy mitochondria. So to do so, researchers collected fibroblasts from cell repositories or collected cells that were donated by the patients themselves. So using somatic cell nuclear transfer, also known as SCNT, for some of the cells, the researchers took them and used this process to sort of fix the already damaged mitochondria using donor oocyte nuclear transfer. So the rest of the cells were induced into pluripotency using factors such as OCT4 and a NOG, which are factors that are necessary and are shown in all pluripotent cells. So to make sure that the iPSCs and the SCNT cells were indeed pluripotent, certain tests had to be done to show this. So one of the tests that they did is called immunocytochemistry, which is basically a test that shows if the cells have the correct surface markers as well as antibodies that are shown in pluripotent cells. Through the immunocytochemistry, antibodies such as PAC6, Nestin, MF20, OCT4, and a NOG were present in the cells. OCT4 is especially necessary because OCT4 is the one factor that is necessary for pluripotency. Another test that they did was a teratoma assay. So basically what they do is that they inject 5 to 6 million of these cells into 8-week-old mice, and after 6 to 7 weeks, the researchers would excise the tumor, sort of section it off, and these tumors histologically showed that they had cells from all 3 germ layers, showing that they were indeed pluripotent. After proving that these cells were indeed pluripotent and stem cells, they made these iPSCs and SCNTs. They differentiated them into fibroblasts, cardiomyocytes, and skeletal muscle cells. After they did this, they would test for ATP turnover and see whether the iPSCs or the SCNT cells would create more ATP than the original, than the cells of a patient with this mitochondrial disease. So when it came to using the cells from patients with certain diseases, the researchers focused on a specific disease called delay disease. This is a neurometabolic disease that affects the central nervous system. So 3 stem cell lines were created from patients that had Leigh disease. So 2 of the lines were induced pluripotent stem cells, so iPSCs, and the third was a somatic cell nuclear transfer line. So what they did was they took these cells and each cell line and they differentiated them into either fibroblasts, cardiomyocytes, or skeletal muscle cells. So they tested the ATP turnover in each of them, and what they found out was that the cells that had been induced into pluripotency through the somatic cell nuclear transfer had a better ATP turnover than the induced pluripotent stem cells because the pluripotent stem cells that were induced had some of the markers that made the mitochondria fail, or that made their oxidative capacity very poor, while the somatic cell nuclear transfer had fixed these mitochondrial defects and had mitochondrial DNA from the oocyte donor. Therefore, they had healthy mitochondria, and when they differentiated, this healthy mitochondria went into the differentiated cells, allowing for a high ATP turnover. From the results, we can see that somatic cell nuclear transfer is way more effective than induced pluripotent stem cells. In this case, just because with somatic cell nuclear transfer, they were able to fix the mitochondria before redifferentiating these cells. With induced pluripotent stem cells, the damaged mitochondria was passed on from the original cell, the original differentiated cell, to the induced pluripotent stem cell, and on to the newly differentiated cells of cardiomyocyte, skeletal muscle, and fibroblasts. From this research, it is possible to create therapeutic stem cell treatments to help treat these diseases, these types of metabolic diseases, especially Leigh's disease. Thank you, and if you have any questions, I'll be happy to answer them.